Barry. Oh, good. Barry Richmond has been coming here for many years, and we go way back. And he was one of the few neurophysiologists who studies, central nervous system, vision, memory. You'll be talking today about working memory, but he was one of the few that took time seriously. This is a true story. Hugo and Weasel got the Nobel Prize right for their work on primary visual cortex, simple cells, complex cells. And once when a famous neurophysiologist, Vernon Mountcastle, visited them, and he said, well, you've discovered all these properties. Well, what's the latency? The time delay between the stimulus and the response. They had never measured it.
Wow, amazing.
But now I think timing is coming in its own, and I think working memory is a particularly good place to think about it. And in fact, that was the theme of my talk. But this is Barry will be telling you about some work he's done. He works on primates, non-human primates, but with many different questions, very interesting results. And in fact, I collaborated with him, one of my graduate students, on actually modeling some of this data, and he may be telling you about some of that today. Okay, Barry.
Yeah, thanks. All right, you guys will have to give me a little leeway. I've never talked about this work either in private or in public in terms of a formal talk. So if I get a little stuck trying to explain something, it's because I haven't practiced.
And by the way, is that the John Wittig that used to be with Prevena?
I was just about to say that. That is the John Wittig who was with Prevena. He decided he didn't want to do chip design. He wanted to do neurophysiology. So he showed up on my doorstep and wanted to study memory. And one of the things I had always said about my lab was I was never going to study memory because it's just a jungle. And it sort of still is kind of a jungle. Even the terminology that I'm going to impose on you tonight is under debate among lots of people. And I'm just going to use the terms the way that I will explain to you and can redo it yourself if you want to. Anyway, John wanted to do memory, and me being flexible said, well, let's do something else. I thought he was doing something else. It turns out he was doing short-term memory. It worked out. Anyway, let me get started. So let me define what I'm going to talk about tonight, which is short-term memory, which is an easier term. It just means you're remembering something for not very long, and we're in ill-defined territory when we try to estimate how long it should be. It's as long as it needs to be and no longer, I guess. But we're going to contrast working memory with familiarity. And the reason will become, I hope, obvious, but to load you guys on what's going to happen. So I'm going to try to convince you that most of the time in our tasks, the monkeys cheat when we think they're doing short-term memory. And what they're really doing is a familiarity judgment. And I'll show you how John very cleverly did the data analysis that makes that seem pretty likely to be true. And then we can discuss maybe at the end whether they ever do short-term, whether they ever do working memory. So I'm gonna say working memory is for remembering details. It has a low capacity. It can be interrupted easily. And we all have the experience of having to remember a telephone number while you walk into the next room to punch the buttons and discover that you've already forgotten it. we've all, at least I've had that experience, I'm sure most people have. Familiarity is the sort of memory you have. If I ask you, gee, you know, I think I saw you once before. And the question is, how good are you at that? Estimating whether you've seen something before or not. And again, I'm gonna try to convince you that monkeys are really exquisitely clever about using familiarity. So if you only want the bottom line, you can leave now.
Is there a threshold difference between low and high and large capacity?
What do you mean by a threshold?
Well, what does low and large mean?
Oh, whether you remember the details or not. Is it a vague memory, which is, I would say, where familiarity falls? Or a memory where you really need to keep track of what the thing you're remembering really is like in detail? And the answer to that is there's no clear separation. But I'll try to show you that we can make it look like there's a separation.
Mhm.
Yes, I have to explain this before I can show you what we actually did. People have tried to study working memory for a long time. And in the 90s, it was a hot topic. And the task everyone used was a task that was actually started in Poland in 1967, and now I've blocked on the guy's name. Think of it in a minute. Anyway, for looking at short-term memory in monkeys, and the task was to show you an object, say A, and then show you another object, say B, and maybe show you another object that's A again. And the job you're to take on is that you will answer no to the one that is not the same and yes to the one that is the same. This is the so-called match to sample task. And it was usually done ABA. The reason I put ABBA up here is what got us started in beginning to understand what the monkeys were really doing was a paper by Earl Miller and Bob Desimone, 1993, in which they thought they were going to make things a little harder and make the sequence ABBA. And it was the monkey's job to ignore the two B's and answer A. What do you think the monkeys did? They answered yes to the second B. And it turned out if you play with this a little bit with the monkeys, what they do is just do whichever one comes along first. And so they're not doing the task that we had hoped they would be doing, which was repeating this thing over what we're going to call distractors. And they just would do ABBA, except it would be ABB. Yes, wrong, but it took Miller and Desimone months to train the monkeys not to do this. And you can do it if you're willing to spend months training them. We knew this. That's what I've just explained. So now I need to tell you what we did. And I'm not going to tell you about all the things we did that didn't work. There were a lot of things that didn't work. But what John did was take a set of stimuli and put them in sequences of various lengths. And the way this would work is the first stimulus in the sequence we would call the trial start and the instruction sequence stimulus. And then we would start showing stimuli, asking the monkey to just look at the set of stimuli. And at some point, we would indicate to the monkey, this is the test stimulus. Your job is to tell us whether the test stimulus matched the first stimulus. We called that match first. Another variation is you could say match any, so that the item you match is anywhere in the sequence. They don't get to answer until you tell them it's the end. But somewhere in the middle, there might have been a repeat, and you can tell them to answer answers to the repeat.
You cannot tell the monkeys.
No, you have to train them to do this. You have to train them to do this.
Right.
And I don't want to bother you
It's
without
an interesting question whether the monkey understands what he has to do.
hang on, okay? We'll see if let's get some data and look at what happened. And see if we can figure out what the monkeys are really doing. Okay?
Okay.
So this is just to familiarize you with the sequences we're gonna look at. There's trial 1, it's one length, trial 2 is another length. We mix lots of trial lengths, get them to do this. And we labeled these match any, which means the monkey could report a match anywhere in the sequence. At the time the monkey was told to
So,
answer.
I mean, are you doing this in blocks or?
Yes, sorry, we're doing this in blocks. Yeah. We tried to do this by instructing, never got it to work. So match any, the sample, the animal answers yes, just to indicate that they've looked at the sample. Match first is that they actually have to wait through the whole sequence and tell you whether it's the first one or not the first one. This was done with sequences of different lengths between sequence 1, in other words, the immediate next stimulus is the test stimulus, or up to 7.
And then there's like some other image that tells the monkey to respond or something.
No, yeah, a spot of light came on to tell the monkey, this is the
test.
trial where you get to try to get something.
Yeah.
It was just a spot of light. Yes,
So is match first, would it be like in trial 1 where you see A and then within the same block, you're matching against the first? Whereas is match any like between trial 2 and trial 3, the instance with the purple?
the reason trial 3 is here is in a minute, I'm gonna show you the monkeys don't even know about the trial structure very well. And they get fooled by stimuli that are actually in the preceding block. You're gonna see data about that.
So you don't have any like blanks or pauses and.
We do have a blank and a pause.
Oh, but-
It doesn't matter. The monkeys are very stubborn about this. Yeah, they do get a little timeout between trials, but it turns out that it doesn't matter. And I'll show you, I'm gonna show you the data and hopefully convince you that that's what they're doing.
How many pictures are there?
Yeah, that'll become another item we'll talk about, but initially we started with a set of 100 stimuli. And we were just picking out of that set. as we got more sophisticated, you'll see that the set size that has a big influence on what's happening. And especially in this, which is same, different. This is put up a stimulus, take it down, put up another stimulus. Is it the same stimulus or is it not, or not? If you do this, you can do this very effectively with only 2 stimuli. And if you do it with 2 stimuli, you can make an estimate that the monkey is actually using some form of real working memory. Because every stimulus is showing up over and over and over again in every trial, or almost every trial. They're showing up. This,
There's only 2 stimuli per trial.
yeah, there are only there are 2 stimuli per trial, and there are 2 stimuli in the set.
There are 3 here though, right?
Yeah, this is, this is to show you what would happen with different sequences. So this to this is no, because you show this and then show this, and then you can show this and show that. That would look like a yes, can look like a yes, and the monkeys do get confused by that. So, and then this,
Are you going to right or left? I'm lost here. Are you doing,
You're going
Which is
top
first?
to bottom.
wait, so you go trial 1, top, bottom, trial 2, top, bottom, trial
Right.
3, top, bottom? Oh, okay.
sorry.
And but when you have a red line, the right answer,
The wrong answer,
the red line is the wrong answer.
yeah,
And the right
the
answer-
red line is answered no. It doesn't match.
Well, the red line, the correct answer is no, and the monkey falsely answers yes because
Yes.
of that association.
But the red line is the false, yes.
Yes.
Gotcha.
Shouldn't-
It takes me a long time to get trained on these things.
And then this is the same thing now running it in a sequence.
Yeah.
This is, the sequence is trial 1 at the left, which is a yes trial. Trial 2 is a longer sequence, but it's a no, it's a yes trial also. This is for match any. And then trial 3, the animal, sorry, this one's correct if you answer yes. This one's wrong if the monkey responds, we think, to this guy in the preceding trial. And we did a lot of this to try to look at that.
I would totally flunk this.
Hmm?
I would do terrible.
Well, we didn't ask you.
I know, I would. Yeah.
So, but the trials are cute, so they know they're in a new trial block, right? There's like.
Well, they're supposed to know.
Supposed to.
Sure, but there is like a queue,
There is a queue,
okay.
yes. But I'll show you in a minute that it wasn't as effective. Well, you'll see. Oh, here, we can actually look at it. So this is where John got creative with the data analysis. In match any, this is the rate of yes response when the sample is somewhere in the sequence of -1 to -7. 0 is the test. So these are, in our terminology, preceding distractors. Except, okay, and so here, one of the first things we found that we just hadn't thought about, frankly, is that the number of the false alarm rate, or the rate, this wouldn't be a false alarm in this case, but the rate, the correct rate was influenced by how far back in the sequence the stimulus was.
The match.
So it meant whatever they were doing, it had some decay-like property, and it looks linear. This is with a 7 distractor set. When we saw this, John did a large number of smaller sets to see what was going on. This is what happened. I think this was this is a set of 3. And then these are images from the preceding trial. So your question about the trial is answered here.
Yeah.
Despite the fact that we put a pause in here and a visual stimulus telling them they're starting a new trial, they didn't start a new trial. This is,
Well, it's much better than this.
But-
It's
Yes.
twice as good. Their
But the
choice is
argument is
good.
it's on the trend line.
yeah,
Wait, what is
Right.
Yeah,
that?
but
What
you
is the
still have
dog
to
here?
compare the -4 here to the -4 here, no? This is just a trial, arbitrary trial line.
right,
So there's still 50%, 40%
yes.
versus
Okay, but
80.
But
they shouldn't
the
have done
trend
it.
line has a
Sure,
different
it's
slope.
maybe
Yeah,
zero,
watch
but
it.
the random rate
Cases
is 20,
for
right? So
Right.
they're 2x worse than they should be, but they're still 2x
Yeah, they're
better
still
than-
doing the task or
Yeah.
doing it sort of.
Can I just, can I
Let
just ask you
me
something?
put one other,
Can I ask you, like, why is this second one different than this second one? Isn't it the same, isn't it up to that?
oh,
point?
it's because, I think it's because there are more intervening stimuli if you have this,
But,
when you go on to the next trial.
but it ought to be easier in the shorter.
I didn't
sequences.
Yeah,
But instead, like that slope decays faster.
right,
So do you have any thoughts about that?
just that's the way it is, and all 3 monkeys did it. With 7 monkeys, sorry.
What is
So
one
each-
dot here?
Yeah,
What is on it?
each dot is a trial.
I'm sorry?
Yeah, what is the dot?
The black dot is a
Black
trial.
dots.
Is this all one monkey? Is this one
It's probably
day?
like a month.
No, this is several days.
Okay,
Scored. Actually, I think this is across monkeys.
I see. But each black dot is a average of a trial of a monkey?
Oh,
on a
oh,
day?
sorry,
Yeah,
sorry.
yeah.
Yeah, it's one data point. It's the set of data from one day,
Got it. Okay, okay.
the average from one
So you
day.
could theoretically
So this
shift
is
them
the
across
scatter.
days.
This is the scatter and they're doing this.
Yeah.
Across a lot of days.
Yeah.
Got it.
And so typically you'd have the same task for the entire day?
Yeah.
Monkey,
Yeah, this was really heavily blocked.
the block is 1 day.
Now, match first, things change. First of all, you get this linear trend again, and it's related to how far you are from the test stimulus. Since this is -1, this is -6, but the monkeys are doing the task. Somewhat. There's some proportion here that we think they're not doing the task. Now, let me insert one other piece of information that influenced what we were interested in.
Wait, so why is the yes rate lower for fewer intervening distractors? Shouldn't that be an easier task?
I'm sorry,
Or am I reading it
He's
wrong?
not incorrect, no. No, incorrect, yes.
It says
it's
yes.
an incorrect yes.
Yeah. That's the open. Oh, it's an incorrect yes.
Yes.
Yes. It's an
Yeah,
incorrect yes.
yeah, exactly.
Pink is an incorrect yes.
Yeah.
Now, the reason this was important to us, I mean, we saw this, all of this was us trying to figure out if we could get the monkeys to do match to sample well enough so that we would be able to do physiology in various parts of the brain that we were interested in. And get some believable data. And when I see something like this, it's a problem. Because some proportion of trials, the monkey's not doing what doesn't appear to be doing what we would like the monkey to do, which is real working memory.
But that's an advantage in neurophysiology. If you had 100% success rate, that's often not great.
often not great,
Yeah, you want.
but if they never do it, it's
Yeah.
not great either.
Sure, but they're doing it.
But I don't know which trials.
But you could predict probably within each trial, you could do like a simple decision variable bound.
Well, we could, and at the time we were doing this, it just didn't seem like it was gonna work.
No, fair enough. It's always, hindsight is easy for us to say.
Yeah, no, I agree with you. We discussed it at great length. But this is what we did. And here, for example, is just a set of 4. You get this extreme case.
So sorry, can you say again, like, why isn't the monkey doing the task?
I'm sorry,
Why do you think the monkey is not doing the task?
I have no idea.
No, but like from the data, what in the data shows us that the monkey isn't doing the task?
Oh, the monkey's doing the task, doing what it thinks is the task. We want it to be doing perfect match in match first. We would like no error or few errors between the sample and the
Yeah.
test.
There's
but
gonna
a
be,
10% error rate is not terrible.
that's not bad, I mean,
No,
there are gonna be mistakes, and it's gonna scale with the number of distractors, so.
10%
It
is
looks
not
all,
bad,
it looks
right?
all perfectly normal to me.
I'm just telling you, look,
Okay.
we weren't satisfied,
Because you wanted the error rate to be lower?
lower.
So what did you do about that?
I'm sorry,
What did you do about it?
nothing. We never figured out a way to fix it. We got distracted.
but the diversity is very useful to compare when they don't do the
Yeah,
task properly and they do, and
You
you.
wanna
can
have
look at
error
the ner-
trials to.
Yeah, like air trails are so
Yeah,
useful in this
to
sense,
see-
If you
Probably
know
ones are
which
different.
ones they are.
but you do know which ones.
Well,
are.
yeah, you know which
You
are
do.
the error trials.
You know what the air trails are, right? So you can always compare the physiology between that. The physiology is super
Well,
useful.
except that if you get an error trial in here. I mean, what we were interested in was, could we see see a signal that was gonna indicate that the monkey was gonna do this correctly? I think that's what we were thinking. And it turned out that wasn't the way we probably should have been thinking about
I
the
see.
problem. At this point, we got the opportunity to do this again with people. Just compare it. to-
Is this data open source?
You know, I have to dig it out. The answer is probably yes.
Okay, that'd be great. I think it's a really cool data set for like teaching decision making. I would love to use those in courses. It's super nice.
We thought we'd compare monkeys and people. We got some. As a side, like, to do this at the NIH meant we would have to get full clinical permission to do this experiment. That would have a danger of maybe boring the subjects to death. But we would have had to go through it. And so we ended up doing this with a group in South Africa. We basically wrote a contract and asked them to do this experiment. And this is what you get. Now, here, you'll see set size down at the bottom. The number of distractors, if that's a relevant parameter. It's not, it is in match first, it's not a relevant parameter in the other two tasks. And how many targets are there? Which in match any can be several, could be several. We didn't we didn't necessarily have only one possible target in the match any condition. So the same stimulus could repeat several times. And this is showing what happens in the same different task. With a large set size, this turns out to be pretty easy for both people and monkeys.
Shouldn't it be harder with a large set size than?
No, it's because when it's a set size of one of two stimuli, one
Yeah.
distractor.
Yeah.
The trace that you have from previous trials is considered to be very strong. And if you have lots of stimuli, it becomes very easy to tell whether you've seen it recently or not. But recency is very hard to do in this situation. Hard to think. And it's considered usually the sine qua non of real working memory. This is just to show the monkeys did the same thing and the people made no mistakes. And here in match first, the monkeys were never as good as the people. The people were good all the time, stayed good all the time. If you decrease the set size, the monkeys have a lot more trouble.
I'm sorry, here each dot is a monkey.
Yes,
So, but what is their variance like? I mean. Yeah, I mean, I guess it'd be nice to know how they do this over learning, right? Because you're doing many, many days and this could change
Yes.
very dramatically. And so maybe humans could just pick it up faster. But by the end, some of the monkeys in the
the
later
monkey,
trials.
the
were better.
humans mostly did it in one session.
But that's because you gave them verbal commands, right?
Yes.
You told them the task. Like if you didn't tell them the task, I'm sure they
Oh,
wouldn't
yeah.
do that.
Right,
So then that would be the right thing to do, though,
it
right?
would be the right thing to do.
Yeah, okay.
It's very difficult to get permission at the NIH to do experiments. That's-
Really
Yes,
interesting.
they apply the full range of clinical guidelines,
Sure.
even if you're doing boring.
Just behavior. Yeah.
Their view is that how do you draw the line?
Sure. I guess you're showing them violent images, maybe, but banana. I'm
Yeah.
not sure.
Anyway,
Yeah, no, I'm just, yeah.
so the monkeys aren't as good as the people. That's the bottom line. And this is match first, match any. The monkeys are as good as the people with the large set size, which was interesting at that point, because at that point, we didn't think we had seen the monkeys do anything that would match what we expected from the people. It turned out it was about the same.
What's your interpretation there?
But match any, they're probably using something like a familiarity or a recency cue. If you've seen it recently. And it turns out they're really good at recency. And turns out people don't like doing this task. They like the match first better. But this is,
To me, match first seems a lot easier, number one thing.
I'm sorry,
To me, match first seems a lot easier. I mean, you remember one thing and you wait for it, while the match any, you have to keep many things in.
right,
So that's very counterintuitive to me.
yes,
Both. I don't know, seven.
Yeah.
right,
It's also interesting, I mean, the humans do a lot worse and the monkeys do about the same. That's,
right.
yeah, that's,
But I mean, maybe another interpretation is like, in the match any, if the monkey kind of, doesn't know or isn't, following the actual, task structure, it makes
Doesn't make
relatively few errors.
they don't know they're doing match first.
Yeah, yeah, exactly, whereas,
Okay.
so.
Do they,
maybe
monkeys
that's
don't know
part
they're
of the
doing
problem.
match first?
No,
Okay, so
I mean,
the humans are told, so,
yeah,
okay. This all makes sense.
Yeah, I think it's just the instructions. I don't know,
Yeah.
That's.
This is-
I mean, how
Yeah.
do
That's good.
how do you
That's
train
good.
them to know that it's match first?
slowly.
Not very well.
But do you increase, how do you train them? Like it's like.
Well, they don't get.
So you start with one or two, you
And
start,
the monkeys train them.
Not very well.
75%.
75% is really good, actually. Given a human can know that you're told there's a trial, there's a structure, you're a thing, this is super trivial. For a
Okay,
monkey, but
okay,
like
gotcha.
the training for the monkey is actually pretty good.
Oh, I see,
So
okay, yeah, that's good.
The question is like, a monkey would need way more time, if you tell the humans this, so I
Right.
think they're actually pretty good.
Yeah.
Yeah,
But I guess. to
That's why that goes
Ellis's
away
point,
when you do the matching. Okay, I
Right,
got you. Okay.
so, yeah, I
Okay,
think that's why
okay.
it goes
Yeah,
away,
yeah,
yeah.
yeah.
And
Okay.
also just the absolute performance for the monkeys and match first and match any is kind of
Similar,
similar.
suggesting that
Yeah.
they're actually doing it very similar.
So doing the same task.
You're doing the same
Basically very
task. Yeah, that
similar.
makes sense. Okay, thanks.
This is just the same issue again.
Barry, did you pay the humans to pay attention?
I'm sorry?
Were the humans paid to pay attention?
No, they were just paid to come to a session and do what they were asked to do. They were guaranteed to get the money,
No matter what they did.
no matter what they did.
Well, you should have made it more competitive.
Well, that was suggested, and that was gonna get us in trouble with the IRB in South Africa.
Oh, it's South Africa.
Yeah, we did these human experiments in South Africa on South African students. Yeah, I knew a guy
Wait a
there
second.
who was
There's
interested in.
people who are actually paid to be competitive, but you're getting competitive gaming.
Yeah, right.
And so, I don't understand
You don't understand.
why anybody would say that making people, enjoy
Oh.
being competitive should be against somehow some ethics.
Well, I can tell you at the NIH, it's exceedingly difficult to get the subjects paid different amounts. They have to know that they're going to get the amount of, how much they're going to get at the session, before the session. So you can't even, you're not allowed to lie to them about what they're gonna get paid or how they're gonna get paid. It's a nightmare.
So you just mistreat the monkeys. Okay, never mind.
Right, I'm gonna skip that. Okay, we have these tasks and I don't even remember how we got into it, but the issue came up, would they be useful in any way in looking at the anatomy underlying working memory? So what we did is have one set of monkeys with the lateral prefrontal removal bilateral. Another set of monkeys with an excitotoxic lesion of the hippocampus bilaterally. And a control set. Okay, so all the data I'm gonna show you now have to do with which part of the brain we're testing on.
So the blue is not the cortex, right?
The blue is
Hippocampus.
hippocampus,
So it's under the cortex.
yeah, it's under
It's
cortex.
a little misleading to paste it on top of the cortex. Not paste, but the color on top. It's okay.
Okay. So here's what happens, non-spatial memory task, which is the task I've been talking about. It's just object memory. When the set size is large, all three groups perform about equally. When the set size is small on same different, we see that the hippocampal animals are somewhat impaired and the lateral prefrontal animals are just devastated. They just can't do this. In fact,
I'm sorry, did they know the task first and then you did the lesions, or you trained them with, they have no
they
experience?
knew the test first.
I see.
That was an issue of which way to do it.
Yeah.
We did it the way that we thought was actually the most contaminated. And we still got the results we got.
Yeah, that's,
And match any, they're approximately equal. And match first, they were roughly the same. I mean, there are quote unquote significant differences, but the differences I think are small. And I don't trust small differences.
What if you do the lesions on humans?
He needs volunteers.
Well, I can volunteer with the PFC.
No, I mean, there are people who do have strokes in those areas, right?
Yeah, but Barry, how long is the delay in time between in this? Barry,
Yeah,
yeah, go back to that plot again, if you don't mind, yeah, so when you say that a set size is 100, it doesn't matter, set size 100, 105 on the right one, say match first. In match first, they have to remember the very first picture they've been shown, right?
right.
And then,
And then there'll be up to 7 pictures shown.
but you don't have that up to 7, right? So you don't know what the rate, you don't know how it changes over time here.
No, we don't. I mean, we have those data.
Okay.
But
Anyway, if there are 2 distractors, then they're all pretty good at it.
Yeah.
Okay. If there are 6, they're pretty bad. Why,
And if they're
2 with a small
so
set
why
size.
is?
A small set size.
Oh,
Yeah.
they have a little more trouble.
Okay, but the last 2 things are about the same, right?
About the same.
Yeah, but
Exactly.
Still, they have to remember the match first for how many seconds? In time?
Oh, it depends how many intervening
But how
stimuli
much
there
you
were
show
gonna
the
be.
picture for, like,
Isn't that what 6 is?
There was, if it was 7, it would have been, I think the trial length was about 100, was about a second here.
So about 7 seconds only,
Yeah,
very short time, really.
yes.
Well, 1 second.
Well, 7 seconds and then the interval indicating that they were moving to a new trial, which I think was a 4-second timeout.
But if the sequence was 7 long, you show them the first picture, you have to remember it then for 7 seconds,
Right,
but they can't do it, really. They suck at
they
it.
have trouble.
They're higher than chance,
No,
right?
They all suck.
These guys are better than chance. They're not so,
No,
they're
but
not
even-
wonderful.
Even the healthy ones kind of suck at it, right? Because their performance drops even over 7 seconds
Right,
to like, well, it's not
Yeah.
chance level, but they have a hard time remembering even. Yeah, for a few seconds,
right,
first picture.
yep. Hang on to that thought. We're gonna see some more of it. So this is what happens with non-spatial memory, like we've been doing. And we thought, well, we ought to do spatial memory. How do we take our task and change it so that it becomes a spatial memory task? What we did was to make a grid, in this case, it was 9 3x3 grid. And the stimulus, sample stimulus was shown in one space of the grid. And the animal's job was to know whether to answer yes or no about the test stimulus being in the same grid hole or in a different one. So it was very closely analogous to the task we were doing before. But now, instead of the cube being an object they had to remember, it's a position in the grid. Is
Same
everyone
picture is
with
fine.
that?
That same picture is fine. Just a spot, they just have to remember.
Yeah, it was I think, yeah, it was a picture, but it was the same one every time. It didn't change for months. Okay, and this is just outlining that you can do the same task we were doing before, except now you're using space as the parameter that you're supposed to remember. The first problem we had is the lateral prefrontal monkeys could not do this. They could not learn anything about space.
How do we see that here?
What are the
The
colors?
red
Go down it.
That's what these three, what these three are.
2.
2. No, there are 3. No,
Oh, yeah,
there is 3.
Same, like colors of different monkeys.
Yeah, they're different. It was so frustrating for the experimenters that we at that point stopped doing a lot of the testing on the lateral prefrontal monkeys. They just didn't. I'll show you what it looks like if they do same different. Here are the hippocampal lesion monkeys. They're really quite good, but hold on to that. Keep that in mind. Working memory, please. And the control monkeys were just about perfect at that test. Turns out that it was much easier to teach the control monkeys this set of tasks than the object tasks we've been doing. They like doing space, position. But this is what the lateral prefrontal monkeys did. That may be no surprise. It's been known for decades that monkeys with lesions of bilateral lesions of frontal cortex have trouble with spatial tasks.
So were these also taught before the region or
They think they were taught after the lesions. Because we did the object thing first. And then did these. Okay. Oh, sorry, Toby?
When you lesion the hippocampus on both sides, isn't it like that patient HM that only had short-term memory? Wasn't that patient, the famous patient that couldn't remember things long-term, always reset every time you had to talk with them? Like, could remember the context of the conversation, but not.
Yeah, the people who believe, yeah, that's a long story, but yes, roughly.
Like that, right? So they can remember over short periods of time, at least the people, at least this patient could remember.
Well, and these monkeys did object memory. Remember, I already showed you that. Or should I go back? Yes,
Yeah, but I think his question has more to do with why is it the hippocampus doesn't have more effect?
well, hang on to that.
Okay.
You're asking us to do this memory task now. Yeah, that's not smart.
We're not being paid.
Okay, this is the same different that I showed you before. Now we're doing match any. Now there's a hippocampal deficit. Monkeys are not having some trouble with this, even the control monkeys. But this difference, I believe, it's not one of these statistically significant, but I don't believe it. This I think is real, and I'll show you in just a second, I hope. If you do match first in space, the hippocampal monkeys are really wiped out. So they can do same different to some extent, but they can't do a longer sequence.
It's weird though, because it's, it's really only like 10 seconds after remember things, right? That's it, and they're focusing on it, right? It's not like they do something else and then you completely forget, like somebody that has dementia. As soon as you change the subject, right, they forget the previous thing. Here they're completely focused on trying to do this. So it's weird. I mean, it seems weird in my casual outsider knowledge. That.
No, it does seem weird. I'm not gonna argue with you. But these are the results, and I think there's no doubt about what the result is here.
Yeah, it's clear.
These monkeys are having trouble with the spatial task. And I think, is it Mackenzie or Alex, one of you has done hippocampal experiments in mice?
I analyzed rat hippocampus data, but I didn't do them myself. We're doing some now, but I haven't done any answers.
But there's been this long debate about whether monkeys have a spatial deficit after damage to the hippocampus.
Yeah.
It's a heated debate because in humans, it seems like it's shown that they have spatial deficits. In rodents, it's shown clearly they have spatial deficits.
Yeah.
It was always claimed that no one could get a spatial deficit with bilateral hippocampal.
But wait a minute, is this a spatial deficit like in the ability to navigate through the world and form place cells and have grid cells? That's a different thing than remember this picture where you have a spot in one spot. That's just a picture. You're showing it a picture and you're asking.
No, you're not. You're showing them a light in one spot.
I know, but it's a picture with a spot somewhere.
You still have to remember the substructure of the picture. That's.
No, but it's still, it's a picture, it's just a picture, it's not the problem of navigating through the world and finding, finding a new map. I don't know if that's a spatial
This
problem.
Buffalo's work and not in this study, it shows the same circuits they use for retinal spatial reasoning as the same, for station.
Yeah, but what I'm saying is, when you put this picture up, it's still a picture and it has a spot somewhere. The spot is the image that you show there, but it's still a picture. You remember this picture, where the things at the top?
Now, all that you need to remember is where it is. You don't need to remember anything about it.
Yeah, but
Yeah,
I'm
but-
just
but-
arguing
but-
that's
but-
just a picture with a
But
spot
Toby,
at the top of
the
the
point
box.
is in hippocampus, when you do a task like this, they're essentially place fields for the spatial locations.
That's been shown.
Yeah.
Yes,
Yeah.
When you do this visual.
Yes.
memory
yes,
task.
yes, exactly, yeah.
Okay.
It doesn't have to be like a physical moving thing, but I see what you're pointing, like, I think that's what you're, like an embodied navigation
Yeah.
task with
It is
this,
a
yeah, yeah.
documentation.
For, one of the puzzles for us, still though, is they're fine on,
Yeah, same different task.
same different tasks, so it's gotta be there's gotta be a little gap in there before you have trouble.
Yeah.
I guess,
Yeah. It's nice, space and time.
anyway, so, thank you guys. These are just some conclusions. Monkeys are heavily biased toward a recency strategy. People use selective attention, which is why they basically don't make mistakes and at first or seem different. Removal of lateral prefrontal in monkeys interferes with all spatial working memory. And these animals couldn't do anything.
So you said that was known before though.
Hmm?
You said that there was evidence that LPC lesions impaired spatial tests, is that right? Didn't you say that?
Yeah.
Yeah. So, so why did you pick those two areas rather than?
Those are two areas that have been implicated in working memory.
Oh, I see, okay. What about parietal lesions? What do you think would happen with that?
I don't predict anymore. I mean, yeah, we do these, this is old fashioned neuropsychology. I hope it shows that it's still of use to the field to know what happens. We're trying to understand how behavior arises from the function of the brain. If you aren't measuring behavior, it's always bothers me a good bit about what is it you think you're measuring? Or why are you measuring it? Anyway, thank you all for listening.
This is beautiful results, but good, go back to your summary slide. I think that you're demeaning monkeys by saying they cheat because they're doing the best they can. Come on.
Yeah, I don't know, if you're really cheating, yeah, it's
It's not really cheating.
not cheating,
Do
actually.
they really understand that it's bad to cheat?
That's right.
Yeah, because I mean, in some sense, what's the risk of them to make a wrong decision? Like,
Yeah.
is there a long timeout punishment or these things? Because like in general, if there's low risk.
Yeah.
You should make errors to test, because maybe the contingency changes, right? Like, we're always probing
Yeah.
the world, so it's,
Or,
they're more feisty, they're not.
Yeah. yes, I mean, like, if you really wanted to kind of train them to respect this kind of structure of the pauses, then like, maybe if they make an error that is consistent with like not respecting.
But they did get a
that boundary,
timeout
then you
when
could
they
give
did
them,
those errors.
oh, you give them an extra timeout for that?
Yeah, they got an extra
But do you get a timeout do they get a timeout for just any error or specifically for that error?
No, you got a timeout for any error.
Yeah,
We
so
can-
that's a.
You might need,
Just that.
you might need some kind of selective
Yeah.
punishment protocol.
Yeah, yeah.
John left before we ever did the physiology, which I'm devastated about.
Yeah.
But someone will have to take it up someday.
Okay, yeah, and
Can I ask you somewhat repeating question?
Sure.
So, can you go back to the results with the non-spatial memory task? Because there you also have the LPFC lesions, but that didn't seem to have as much of an impact, is that correct?
Oh, the non-spatial,
Right, so in the beginning, we get.
Here,
Here,
No, this one had a big effect, but the other ones, they're.
Which one?
Oh, so this one still had a big effect,
Yeah.
okay,
yeah,
cool, okay. Do you think this has any relationship with? There was a Earl Miller paper from a few years ago talking about traveling waves in prefrontal cortex during a delayed match to sample pass. And they basically show the waves change direction and they increase in wavelength and frequency.
yeah, what happened? Now, are you interested in it in normal animals or animals with inflammation?
I guess,
Because I don't know what happens to traveling waves when you ablate the tissue you're looking at.
right,
But they're only traveling, that's for sure.
yes.
Yeah, because there's no tissue, but what happens to the tissue in the surrounding areas? In fact, that was something I thought of during your talk.
Right,
Yeah, are you disturbing that structure
Right.
throughout the brain, even though you've done what you consider to be a selective interference?
Yeah.
Is that roughly what you're?
I was thinking more if for some reason these type of like relatively short-term delay match to sample tasks, that memory is being stored in a traveling wave in prefrontal cortex that would match up with your data and it would match up with their data. Those settings.
I can't say, it looks like it matches up. Yeah, you'd have to do something specific to sort it out.
Yeah, try to decode from the waveform.
so how did the size of the lesion correlate with their performance? Because there's like one monkey here.
Yes, we always get that question, and the answer is never very well, and I can't tell you in these animals.
Okay,
I can tell you with these kinds of lesions, it's usually a frustrating
yeah,
enterprise to
Okay.
try to chase the individual animal's performance against the size of the lesion.
okay. Well, what do you think of this one monkey that didn't really have any deficits beyond that would roughly be expected? Yeah, the red and the, yeah.
Yeah. Did we miss some tissue? Maybe.
Yeah, no, that says not, or, yeah.
And in fact, we're trying to finish writing some papers about this.
Yeah.
Well, this is, believe it or not, probably the beginnings of this are a decade old.
Sure. Oh, no, I understand that.
I've never had the courage to talk about it before. I don't know why. What I can say is Terry had a graduate student who did his PhD modeling the basic task, not the deficits, basic
Okay.
task.
Yeah.
And came up with a very nice hypothesis of what's going on, arguing that you're going through a sequence.
well, he trained a recurrent network basically on these tasks.
I see, but
Yeah.
what about like a baseline POM-DP or some Markov decision process?
No, we didn't do that. But go ahead with the summary.
Well, anyway, he did it, and what it looked like is the more practice you gave the network, better it did on match first. Looked like it went through a progression
Hmm,
from match any to match first.
okay,
And it was dependent on the parameters of the simulation, not exactly how quickly that happened.
sure.
But he raised the possibility that it might be that you're looking at some maybe emergent property that happens with more training and a larger network. I mean, because one of the things that always bothers me is why should we expect, for instance, tasks like this for monkeys to ever do it as well as we do?
Sure, yeah,
We got a much bigger bit of wetware to do this.
yeah,
I mean, I always like to point out that we think of the frontal cortex as a piece of tissue that has to do with planning and looking ahead. And you're trying to compare what a monkey does compared to a human.
Mhm.
When was the last time you had a monkey discuss with you their retirement benefits?
yeah, sure, yeah.
You know, just
I've never met a monkey, so I can't say for sure, but I'm pretty confident they wouldn't.
Yeah.
Okay, well, thank you, Barry. Yeah, yeah, I think he gets the prize for being on time.
Yeah.